All forms of transport have to do with the plasma membranes. Phospholipids make up the plasma membrane and are composed of one hydrophilic head, which contains a phosphate group, and two hydrophobic tails, one being completely surrounded by hydrogens and straight, which is saturated, and the other being bent and not completely surrounded by hydrogens, which is unsaturated. These phospholipids bind and together form the plasma membrane. Diffusion is the movement of cells through a plasma membrane from a high concentration to a low concentration. This requires no energy because it moves across the concentration gradient, which is from high to low concentration, meaning it is a form of passive transport. It does this until equilibrium is reached, which is when both solutions have equal concentrations. It is easy for nonpolar molecules to move across the membrane because they can directly go through the plasma membrane through the nonpolar region of the fatty acid tails. But polar molecules cannot do this without the help of a transport protein because the nonpolar fatty acid tails repel them. The use of a protein for molecules of ions to go through the membrane is called facilitated diffusion. The first type of protein that can be used is a channel protein. This protein has a tunnel that goes through it that allows the polar molecules or ions to go through it. The second type of protein is a ion-gated channel protein. A molecule has to bind to the protein to raise the lid of the protein, thus allowing polar molecules or ions to go through it. The final type of protein is a carrier protein. A polar molecule or ion will bind inside of the protein, and the protein will change its shape to allow the molecule or ion to pass through the plasma membrane. Osmosis is a form of facilitated diffusion. It is the movement of water molecules through a membrane with the help of a protein called an aquaporin from a high water concentration to a low water concentration until equilibrium is reached. Basic outside structure of a plant consists of a cell wall and a plasma membrane. The basic outside structure of an animal cell consists of a plasma membrane. Tonicity is the ability of gaining or losing water by a cell, and it can happen in three different environments: isotonic, hypertonic, and hypotonic. A hypotonic solution is the best for a plant cell because the water coming into the cell makes the cell swell, but the cell wall stops the cell from bursting, allowing the plant to be strong and turgid. An isotonic solution is not ideal because water comes in and out of the cell at equal rates, making the cell lack turgor pressure, which is the pressure exerted by the cell wall on the water coming into the cell, so it becomes flaccid and weak. In a hypertonic solution, the water leaves the cell faster than it comes in, so plasmolysis occurs. This is when the plasma membrane separates from the cell wall and it kills the cell. An isotonic environment is the preferred solution of an animal cell because water goes in and out of it at equal rates. The cell is at equilibrium. In a hypertonic solution for an animal cell, the water concentration is greater inside the cell, so water will leave the cell into the solution until equilibrium is reached. This will dry out the cell and it could possibly die. In a hypotonic solution for an animal cell, the water concentration is greater outside the cell, so water will come in the cell until equilibrium is reached. The cell will swell and eventually lyse, which means burst. Active transport is the molecules or ions are pumped from a low concentration to a high concentration across a membrane until equilibrium against the concentration gradient. This requires energy in form of ATP. An example of this is the sodium ion and potassium ion pump. The sodium ion concentration is greater outside the cell, so 3, represented in green, will go from the cytoplasm into the protein pump and get released into the extracellular fluid. Then 2 potassium ions come into the protein pump from the extracellular fluid into the cytoplasm. This process repeats. Bulk transport also requires energy, and the first form is endocytosis. Endocytosis is a process of the cell taking in nutrients by engulfing those nutrients from the enfoldings and plasma membrane. The first form of endocytosis is phagocytosis, which is when the plasma membrane engulfs the surrounding food particle in the extracellular fluid. This forms a food vacuole inside the cell. The second form is pinocytosis, which is when extracellular fluid is engulfed by a cell in order to get solutes. The cell doesn't want the fluid, but it wants the molecules dissolved inside of the fluid. This forms a vesicle. The final form of endocytosis is a receptor-mediated endocytosis. A specific molecule called a ligand will bind to a receptor on the plasma membrane. These receptors with the ligands will congregate in one area on the plasma membrane shown in the drawing and get engulfed by the plasma membrane. This forms a coated pit. After the receptors are completely engulfed, they form a coated vesicle. The other form of bulk transport is exocytosis, which is where a cell gets rid of waste through a vesicle. The vesicle leaves the endoplasmic reticulum and goes to the Golgi and fuses with the plasma membrane. The vesicle then releases its content into the extracellular fluid. This is only done by single-cell organisms.